capable of doing, right? Which is excitability, contractility, elasticity, and extensibility. Memorize those words, and by the time we are done with Monday, we're going to come back to these words and say, see, this is what we meant by excitability, right? For right now, it means to being able to create an electrical signal, an impulse. I'm going to have to walk you through how that actually happens. If something contracts, what is it doing? Getting smaller, yeah. So muscles can get smaller, they can contract. And we'll learn how they do that and how much force they're capable of producing when they do this. But I think if it's elastic, what does it have the ability to do? Just stretch. But when things stretch, what do they end up doing sometimes? Recoiling back, right? So elasticity and then extensibility. I can pull it and stretch it, but it's really the elasticity that gives the ability to recoil and go back to its original size. And we'll look at why all of these things are possible. But for right now, it might be just memorizing 4 different terms that you're not quite sure really how they relate to muscle tissue, but you've memorized the terms, so you talk about them, you can start putting the 2 ideas together. Why aren't you working? Come on. What is wrong with you? Talk. Yeah, I know you did that. Thank you. All right. So, pop quiz. He's like, dude, you just turned it off. What's the name of the cell that produces protein fibers in the extracellular matrix of connective tissue? Fibroblast. What's the name of the cell that produces cartilage? What's the name of the cell that produces bone tissue? So what would you guess would be the name of the cell that produces muscles? Something blast, right? Right? And myo refers to muscle, right? So myoblasts are the cells that create muscle tissue. But what's interesting is from mesenchyme, right, embryonic connective tissue, embryonic stem cells, they're going to turn into myoblasts, and they fuse together, right? So those guys start to fuse into one. And what's nice is that whatever they have, they contribute to the growing structure. So they start adding the organelles, they're adding the nucleuses. So when you look at a growing muscle cell, which is what this is, we refer to them as being multinucleated. Because each myoblast, when it joins the party, right, it gets bigger and bigger, right, contributes its nucleus to the growing overall structure. Now this is how we form a muscle fiber, right, by the joining of multiple myoblasts. And what you have to realize is a muscle fiber is a muscle cell. It's the same thing. And just like when we talked about in terms of intramembraneous ossification, where those last mesenchyme cells didn't join the party to actually make the bone tissue, but they ended up being the periosteum on the outside. Well, myoblasts that don't become a part of the growing muscle fiber stay on the peripheral edge and act as stem cells. So when you damage your muscle tissue, they can become activated and start making more myoblasts and start adding and repairing the damaged muscle fiber. So here's a photograph of an actual muscle fiber, an actual muscle cell, and all the nuclei, right, that came from the myoblasts. Now we got to go from macro to micro, from big scale to small scale. We're going to go down to the molecular level with this. So, big scale. Out of curiosity, what bone might this be right here? The humerus, all right. So now what I have is maybe your biceps, right? The muscle. And I've already asked, well, what is a muscle comprised of? Well, really, this big muscle is comprised of bundles of fascicles. All right, so a muscle is a collection of fascicles. I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, maybe 11, 12 fascicles here. Well, how do I bundle all the fascicles up into one big pile? Well, we're using a type of connective tissue called epimysium. It's wrapping around all of the fascicles. All right, well, how do I keep one fascicle separate from its neighboring fascicle? Well, let's wrap it by a layer of tissue called the perimysium. So perimysium is wrapping around each individual fascicle, but all the fascicles are bundled together by the epimysium. It's the same tissue. It just invaginates inward. We change the name so we know what it's wrapping around. And if I realize that fascicles really are collections of these little circular things. Well, what are these little circular things? Let's pull one of them out. Oh, what is it? It's a muscle fiber. What's the name for a muscle fiber? A muscle cell. So really, a fascicle is a collection of muscle cells. Well, how do I bundle up my muscle cells? Well, right, they're really within a fascicle, so I'm wrapping all the muscle cells up with that perimysium. So that's what a fascicle is, just a bundle of muscle cells. But what is each muscle cell wrapped with? So I can keep this muscle cell separate from that one or this one or that one. It's a layer of tissue called endomysium. So epimysium wraps all the fascicles up. Perimysium, right, is wrapping the individual fascicles, which really is a collection of muscle fibers. And then the endomysium is wrapping around the individual muscle cells. So it's sort of like taking a box of packaged spaghetti noodles. Right? The box could be the epimysium. Each individual wrapping of spaghetti package, right, is a perimysium that you have to break open to get the noodles out. But you need to wrap each noodle with saran wrap. Right? So now you've got an individual spaghetti noodle wrapped with saran wrap, bundle them up, put them in a grocery bag of plastic, and put all those bags of spaghetti noodles into a box. But if you look at this, right, it's really just bundles of this to create that, and bundles of this. We got to get down to that level, and then the little teeny dots you can barely see right there. So let's look at it from a different perspective. Right? This is a fascicle, right, which is then one of these things right here, or one of those. So what's a fascicle made up of? Well, it's made up of bundles of muscle fibers, which is AKA a muscle cell. And how do I bundle up all my muscle cells? I wrap them all up with a layer of perimysium. And then I realize that each muscle cell is surrounded by a layer of tissue again, right, called the endomysium. Come down here, we have a muscle fiber. That's one of these things. But remember, a muscle fiber is AKA what? Muscle cell. Those cells have membranes? Yeah, they do. So when you peel off the endomysium, which was the layer surrounding a single muscle cell, you actually get underneath of it this little semi-transparent layer you can barely see, but they're showing it to you right there. Right? That's the sarcolemma. That's the name we give the membrane to a muscle cell. And we're going to change that. So lemma refers to membrane. We'll have a neural lemma when we get to the nervous system. But sarco is just the membrane of a muscle cell. Remember, all cells have cytoplasm? Well, we call that fluid the sarcoplasm. That's just the cytoplasm of a muscle cell. You want to get used to the repeating theme that we see here. If I go back to the overall idea of a muscle and what it is, right? It's bundles of fascicles, these circular things, but then that circular thing seems to be a bundle of other circular things. And then I realized that those bundles are yet bundles of other circular things. Now we're just getting smaller and smaller and smaller. Do you guys know the Russian dolls? Mm-hmm. Right? Where you get a smaller doll and inside of a smaller one and a smaller one still. And that's what we're doing Russian dolls with muscle tissue. And this is where we want to pause. This is where I tell you guys stop right here. Get used to this level of organization first. Muscles are collections of fascicles. Fascicles are collections of muscle cells. And apparently, muscle cells are collections of something else that look like little rod-like things. What are they called? They're called myofibrils. Warning, Will Robinson. What's the name of a muscle cell? A muscle fiber, right? Otherwise known as a myofiber. Muscle fiber is a myofiber, but myofibers are collections of myofibrils. You gotta get the brill in there. You gotta roll your R's maybe, right? So a muscle fiber, a myofiber, is a collection of myofibrils. Well, here's another way of looking at it. It's from a different book. But it's the same idea with a little more artsiness to it. This whole thing is a single muscle cell. All right, I know muscle cells have membranes. I know the membrane's called the sarcolemma. Well, that's what this gray sheet is. That gray sheet is the sarcolemma. That's the membrane to this muscle cell. I can then see, right, this little structure here, and that structure here, and that one. These are all the myofibrils, right? So these things here are those things right there. All right, now when I look at it, I realize that each one of my myofibrils apparently is wrapped by a bluish mesh looking like material. And it runs along the entire length of those myofibrils. Well, that bluish mesh looking like stuff is called sarcoplasmic reticulum. It's ER from a normal cell. Remember guys who had rough ER and smooth ER? Remember what the function of smooth ER was? Making lipids, right? And storage of calcium ions. Well, this is endoplasmic reticulum, but it's wrapping around the entire myofibril, around the entire length, right? So let's make my arm, right, a myofibril. My sleeve is the endoplasmic reticulum, totally wrapping around the entire length, okay? Now, go back to the gray sheet. It's a cell membrane known as the sarcolemma. What are cell membranes composed of? A phospholipid bilayer, right? So when you see this gray sheet, you're thinking that idea in your mind, right? It is a phospholipid bilayer. Now these purple things, if I was an ant, my little red light's an ant, I'm walking along the top of the membrane, I could fall into that tube. Well, that tube is called a transverse tubule. It is just an extension of the cell membrane, but it forms a tube, a tunnel. Which means, what is the transverse tubule comprised of? A phospholipid bilayer in a tube-like structure. Same material. Now, where does this tube go? Well, it seems to be going into the cell, right? Into the cell, but it's it's also wrapping around myofibrils, but not as completely as encompassingly as the endoplasmic reticulum. So if my forearm is a myofibril and my shirt sleeve is the blue endoplasmic reticulum, this is going to be my transverse tubule, right? And it's wrapping around maybe here, right? Coming from, don't die over there, right? Coming from the cell membrane. Whoops, come here. Hey, stay together. All right. Coming from the cell membrane, but it's here, and then it's going to be here, right? And it's going to be here. These little tube-like structures are wrapping all the way around the myofibrils. Sort of like your belt goes around your waist. And since they are an extension of the cell membrane, I know their structure. They are a phospholipid bilayer. Now, I've got my meshy stuff here, but the ends of the mesh look like little blue tubes because it's a sac-like structure of this mesh looking like material. So the ends of the endoplasmic reticulum, we call them lateral sacs. Lateral for side, and it's a sac-like structure. In there, your muscle cell will store the majority of its calcium ions. All right? The bulk of your calcium is in those lateral sacs. Now, to help you with practicing some of the organization of muscles are collections of fascicles. Fascicles are bundles of myofibers, and myofibers are bundles of myofibrils. You have this little model, all right, to help you kind of tangibly see this idea with this. Now, we still have to explain, you see the large dots and the small dots? You got to get to those things. So, different book, different way of representing this stuff is this. Because the more different ways you see things, the more we can understand them. First, read the title. That's a muscle fiber. And that means what? It's a muscle cell. Okay, that makes sense. So there they're peeling back the sarcolemma. got that, which means that the endomycelium is gone, right? They peeled that off. They peeled this off, right? All they're showing me is this right here. The endomycelium is gone, all right? They unwrap it. I can see all the individual tube-like structures called myofibrils. I can see now they changed color, the purple mesh-like material, and then the green, which are the transverse tubules. And here's a side view of one entire myofibril. Right? There's the sarcoplasmic reticulum, and if you look right here, there's the sac-like purple part with the green tube, another sac-like purple part. When you have the purple, green, purple, it's kind of hard to see at that distance where you guys are, but it's this idea, right? Blue, purple, blue. You get these three structures together, right? Two lateral sacs separated by a transverse tubule. That's what you have over here in this image, right? Now the transverse tubule is the green part. I've got two purplish tubes on either side of the green. That's called a triad. So triads are comprised of a transverse tubule flanked by two lateral sacs. All right, and that'll become really important to how we get calcium to come out of these guys a bit later. Down here they're showing me, what are they showing me? They're showing me sarcolemma and T-tubules. Well, there's the sarcolemma, and they're giving you the vision that it is a phospholipid bilayer, and that bilayer dives down into the guts of the muscle cell, creating this tube-like structure called the transverse tubule. So now you can visually see that the transverse tubule, this green part, is comprised of another phospholipid, or actually the same phospholipid bilayer. And then we have these protein channels that are in there, and they're called transporters. And you guys by Wednesday need to understand where there's more sodium, more potassium, which way the ions are going to diffuse, why they're able to diffuse, the idea of simple diffusion compared to facilitated diffusion, all these things that I'm hoping and wishing you were taught in cell biology, right, which I take for granted you actually were. So know that list, right? We'll come back to that topic in a second. All right, let's zoom in a little more microscopically. All right, we've gone from muscles, which are collections of fascicles, collections of myofibers, muscle cells. Muscle cells are collections of myofibrils. But now myofibrils are collections of these two right here. Right? The idea of myofilaments. We have two different types of myofilaments. You have actin myofilaments, because they're comprised of the protein called actin. And we have the myosin myofilaments, because they're comprised of the protein called myosin. Myofilaments may sound familiar if you remember the structure of a cytoskeleton. Right? Myofilaments, intermediate filaments, and microtubules. And if you remember that myofilaments, right, the myosins are motor proteins. So this is going to be like a little motor protein-like structure that we're going to be looking at. How are they designed? Well, let's get really tiny. This is an actin myofilament. This whole thing right here. All right, that's going to be the red ones up here. All right, see how they're smaller than the gold ones. So these are the thin, those are the thick. But I want you to know the actin myofilament name. Know that name, not so much the nickname of thin filaments. What is an actin myofilament comprised of? Three things. First in your mind, envision two strands of red pearls. Right? And now twist the red pearls together. Those are my strands of actin beads. I've got two strands of spiral actin proteins, and then wrapped on top of those two strands of actin proteins are two strands of these green rope cable looking like proteins. There's actually two of them. This one comes here, goes behind the actin, you can't see it, then it pops out again over here. So this strand is not the same as this one. These are two separate strands called tropomyosins. So in my mind, I've got two strands of red pearls and then two strands of gold necklaces. Got them off of George. Okay? Now, I'm going to have these little pockets of proteins called troponin complexes. Those are the three parts, right? We've got actin strands, tropomyosin strands, and a troponin complex. They make up your actin myofilament. Now, to help you in the future, a little terminology. Whenever you see the word complex, don't think of it as complicated. Think of it like a building complex, right? Multiple buildings to the overall structure. This is a multiple part thing called troponin. How many parts to the troponin complex do you think? You got 3. I see 3 little blue circles. All right, right? There's 1, 2, 3. There is another troponin down there. So, of these 3 parts, each one has a name. I could care less with the name. But what they do, I want you to know. So, one of them is permanently attached to the actin protein, to the actin bead. One is permanently attached to an actin protein. The other one is permanently hanging on to a tropomyosin strand. So the other one is hanging on to a tropomyosin strand. The third one acts as a calcium binding site for the calcium ion to land on. So one part is a calcium binding site, one part binds to tropomyosin, the other part binds to actin. And we're going to see this little idea a couple of times, not so much tonight, but on Wednesday and next Monday. Now, if one of them is a calcium binding site for the calcium ion, there's a basic rule you want to learn because it'll help you through all 241, 242. A basic biochemistry concept. When a chemical binds to a protein, the protein will change its shape. Memorize that. Make it your mantra. Right? When chemicals bind to proteins, proteins change their shape. And the same is true for when the chemical comes off of the protein. It'll change its shape again. Now, let me give you one example you might be somewhat familiar with. There's a protein in your blood that carries oxygen. What's it called? Hemoglobin, right? Hemoglobin changes its shape for every oxygen that attaches to it. And when the O2 comes off, it changes its shape. And that changes its ability to, or desire to hang on to additional oxygen molecules. Who here has had organic chemistry? Remember cis and trans? That's what this is, all right? So just understand, keep in the back of your head, that whenever a chemical lands on a protein, that protein needs to change its shape. When the chemical comes off, the protein goes back to its original shape. That'll help you through all 241 and 242. Whenever you hear the word complex, that means it's a multiple part structure. Not that it's difficult, all right? It could be, but it's the idea of multiple parts. So that means that my actin myofilament is comprised of these 3 protein structures, right? 2 strands of actin beads, 2 strands of tropomyosin cable-like structures, and then placed at regular intervals along the length of the actin beads are these troponin complexes. Now, myosin's a lot easier.
I have a question about there's 3, but the one hiding in the back, is that another complex?
Yep, it is.
So it's
another one.
Another group of 3 probably. Okay.
Exactly. Now, to help you make sense, we'll get to that in a second. This one's easier, right? Myosin, myofilament. Think of it as either one of 2 things. A Dr. Seuss hat rack for lots of hats, right? But each myosin is a 2-headed structure. There's a long tail. Think of it as a 2-headed golf club that you just shove in a golf bag, but all the different clubs are at various lengths. So when you look at the length of a myosin myofilament, it'll look somewhat like this. Each one of these is going to be 2 heads, all right? I can't make it individual head. 2 heads here, 2 heads here, all right? 2 heads here, 2 heads here, 2 heads here, and 2 heads there, right? So imagine my arm is a styrofoam arm, and you stick popsicles in my arm, right? In a radius-like area around my arm, right? So this is like a porcupine coming like this. Along each one of these is going to be an actin strand. So this head can grab this actin, this head can grab this actin. So anybody along this line grab the same actin strand. Anybody along this line grabs a different but the same actin strand. So these are myosin heads. To help you with that, because it's sometimes hard to visualize, is this little friend. What did you guys pay for this? $5. That's what I paid for it.
150.
Gotta go higher, dude.
300.
Gotta go higher.
Grand.
Bingo.
Yeah.
Be careful with it. Be careful with it. But, right, what it's showing you is the actins. See the little green things? And the yellow? Trypomyosin, troponin complexes. And we can pull it apart. Those are your myosins and the little club heads. So when you look at it, you can see how one blue cable-like structure, right, has all these heads that go all the way around. So a row of actin here, a row of actin there, as you can see on this side. A row of myosins. So this whole thing is actually a model that we're going to call a sarcomere, right, which is the next slide. That's this, right? So this thing is that thing, right? This is this. That empty space is this empty space. This red Z line is that red Z line right there. Now, I'm pissed because for $1,000, I want my money's worth. When you look at the sarcomere, and you guys need to do this, first of all, we will define a sarcomere as going from one Z line to the other Z line, right? That is the boundary of one sarcomere. And I realize that the red line is my actin, and the blue ones are my myosins. And after careful studying, I realize that the actin is physically connected to the Z line every single time, on this side and on that side. Is that true for the myosins? No? Nope. What are myosins connected to? A little green squiggly thing, right? What's a little green squiggly thing? Oh, a protein called titin. It's an elastin protein. It's stretchy. So I realize that by looking at this, that titins connect to the ends of the myosins. Myosins don't connect to the ends of the Z line. It's the titins, like the intermediate, the middleman. It connects on one side to the Z line, and to the other side it connects to a myosin end. And it does that on both sides. Along the middle of my myosin is no man's land. There are no heads there. It's bald. Now, let's try a little mental exercise. In your mind, I want you to look at this Z line and picture it and staple it to that position so it won't move. Over here, though, I'm going to grab the Z line like an accordion and pull it this way. Can you see how I'm pulling the red lines along with it? Am I stretching out the green lines? Right? We are then uncoiling this elastic green protein. When it becomes straight, maybe it begins to then pull on the myosin, and then as the myosins move this way, I begin to straighten out that green line. But they're all made of elastic protein. So maybe I take the Z line, that side doesn't move, that Z line, but I bring it all the way to here. If I let go, what's going to happen? It's going to zoom right back, right? That is elasticity. So the elasticity is due to the titin proteins that connect the ends of myosins to the Z lines in a sarcomere. But the sarcomere is also the functional unit of your muscle. Whenever you do anything, any kind of a force you create, this is the workhorse. This is what's creating the force. Every myosin head, like a person on a tug-of-war team, grabs the actin and pulls on it. That's what a muscle contraction is. And that's our job on Wednesday, to teach you how this happens. So if I think of every little head as a member of a tug-of-war team, and the red actin is my rope, you want to keep in mind how many of my team members are adjacent to the actin strand. I look over here and I realize I've got some sluffers here. These guys aren't next to the rope, which means they can't be a part of the force production because there's nothing for them to grab. And we're going to be looking at the relationship between how many of my myosin heads are adjacent to the actin compared to the amount of force I can create. You guys with me so far? No. All right. No. So where did you go bye-bye? Oh, about 35 minutes ago. Where did we lose you? It's okay to get lost. You have an idea of what actin's comprised of? Okay, let you work on it, but remember, this is a nice physical, tangible model, right? To a relationship between the red things, which are actin, the blue things are myosins. You can see the troponin complexes, those little green things on here, right? It's $1,000, so don't drop it. And then what about this? If this is a part of a myofibril, what are myofibrils wrapped with? That's a myofibril. They're wrapped by sarcolemma reticulum, right, and transverse tubules. The gold is a transverse tubule. The green is sarcolemma reticulum. You can see how it's wrapping around the entire myofibril.
And calcium is in both of those?
Calcium it's in all of it, but it's mainly in the lateral stacks. Okay. That's what we're going to get to come out of. We talk about this on Wednesday. Okay? So, when this, which is a myofibril, not a myofiber, this is a myofibril. This is a sarcomere. Right? Look at the distance between the two red lines. Right? See, that's a lot shorter here. Because when the myosin heads grab the actin on this side, these myosin heads grab these actins, they're going to pull the Z line this way on this side. But these myosin heads grab those actins and pull it this way. So we're taking our sarcomere like this, and we're shortening it, and then relaxing. Contracting and relaxing. Relaxed and contracted state. What you're noticing is that if this is a myofibril, then I've got multiple sarcomeres along the length of a single myofibril.
Does the contraction happen in the sarcomere on both sides all the time, or could do it?
No, so it's, here's the next idea. Whenever a muscle cell works, that means all the myofibrils in the muscle cell work. That means all of the sarcomeres along all the myofibrils work as well. It's called the all or none principle. Either everybody in the muscle cell works or nobody works. And a part of our goal on Monday of next week perhaps is, if I were to take my finger and do this, like maybe like in the matrix, right, where I kind of do this, right? I'm getting a certain number of muscle cells to work. But whatever muscle cells are working, every sarcomere in those muscle cells are beginning to shorten. The only question is by how much of their ability? Is it 1/10 of 1% or 100%? Because this is more of a contraction of the same sarcomeres as this is. Same number of sarcomeres, just not to their potential. We're going to see how that works on probably Monday of next week. So let's get one last little topic down. I know my brain controls my muscles, and we'll teach you how that works, unfortunately, toward the end of the quarter. But the thing that talks to your muscle cells are motor neurons. So I've got a motor neuron, right? And then it's going to split. And what we're looking at is, for that one motor neuron, how many muscle cells does it connect to, right? And they're showing you two different motor neurons, the gray ones and the orange ones. So apparently the orange motor neuron is going to 1, 2, 3, 4 different muscle cells. The gray motor neuron only goes to 2 muscle cells. So when you look at a motor neuron, right, and all the fibers it connects to, that's what a motor unit is. A motor unit is defined as a motor neuron and all of the muscle cells, that's what the fibers are, right? All the muscle cells it connects to.
So that's 2 different motor units on the top.
Yep, exactly. Now, the place it actually connects to, right? There's a photograph of it, right? There's a cartoon of it. the actual place where the motor neuron connects to the muscle cell is called a neuromuscular junction. That is the physical place where the motor neuron comes in, quote, contact with the muscle's membrane, the muscle cell. All right, so who gives a flying flip about motor units? Well, let's look at, I think actually it was the very first question in your exam tonight. The relationship idea between stability and mobility, right? We have a relationship here. Now, which motor unit has more muscle cells in it, the gray or the orange? The orange, right? So the larger the motor unit, the more force you can create. Because you're activating more muscle cells, which means you're activating more sarcomeres. Right? So the larger the motor unit, the more power you can create. But you got to sacrifice something for that power. You have to sacrifice control. Just like for stability and mobility, right? The more stable you have, the less mobility you have. The more power I can create, the less control I have of that power. What's the opposite of that? Less power. So in other words, the smaller the motor unit, right? The less power I create, but the more control I have. Does anybody here play a stringed instrument? It's like, yeah, I'm not raising my hand. What do you play? That's the second person who plays. Okay, great. All right, so you have your bow arm and you have your finger hand, all right, along the fingerboard. To be able to do this requires a lot of dexterity, doesn't it? A lot of fine control. Do I want a large or small motor unit to do this? You want small. If I want to try and kick a 65-yard field goal to win the football game, do I want a large motor unit or a small motor unit? How many field goal kickers get booed at when they miss by just that much? It just went to the right a little bit too much, all right? Because the more power you create, what do you sacrifice? You sacrifice control. And you learn this as a little kiddo. If you have any toddlers to be around, especially little ones are learning how to grab things for the very first time, you watch a little kid grab a plastic drinking cup, right? And he might be okay the first time, but it's empty, right? But then he sees the cup and his brain goes, oh, that's my cup, okay. But then there's milk in it or juice. And he's never picked it up before with juice in it. So it weighs differently. And he's using the same motor he did the very first time, and he ends up dropping it because his brain went, holy shit, this is heavier than before. I need to activate more motor units or a bigger one because I don't want to drop my juice. And so you learn that when you see that object, it's going to weigh a certain amount right off the bat. And you learn this, right? You guys now, if you were to go up to a 100-pound bag of flour, right, you know it's going to be a heavy thing to lift. You don't just lightly pick it up with a cup of coffee, right? You kind of scrunch down and you pull it and then you lift. But if you saw an empty bag of flour, he'd probably use 2 fingers to pick it up because his brain realizes, oh, I've learned by experience that that doesn't weigh very much. I don't need to use a large motor unit. You always start off with the smallest motor unit first. And your brain rapidly increases it to the appropriate size. Because the small one's a guiding, like a tugboat. Right? It gets your hand to the object. Then I realize, oh, it's a bunch of paper towels. It probably weighs ½ pound. I need X motor unit to lift it up. If I've never seen one before, then it's a little bit of a mystery. So I used to screw around with my UW students. We'd have lab. I'd have a bunch of coffee mugs. And they're all empty. And their task is to estimate the gram weight of the mugs. Okay, great. Go to break it. Get the hell out of here. Go on a break. And I swap them out for lead-filled mugs. And they say, okay, guys, we need repeatability. All good scientists repeat their results. Let's do it again. And almost everybody drops them. Because their brain said, oh, this is easy. It's only going to weigh 15 grams. When it weighed a couple of pounds, actually. All right? And now those that have really fast reflexes, it begins to fall, but then they grab it really quickly. Because the brain said, holy shit, we need to increase the motor units because it's going to fall if we don't do that really quickly. And that's what your cerebellum does. begins to realize how much things are going to weigh and how you get to operate all these things. We're almost done, almost done. All right, this. What I'm trying to teach you guys to do is to don't look at a picture, but look at the picture. Get your bearings. What are they showing me? Where am I? Right? I realize that it's a membrane because I can see the cartoon of a phospholipid bilayer. Then I realize that this is a motor neuron, and this is the membrane then of my muscle cell. So this is a cartoon of what that looks like right there. I've got all these transport proteins in the membrane. I've got ions of sodium, potassium inside and outside. And don't forget, this is also a membrane. It is another phospholipid bilayer. They just took the shortcut and didn't show you it like they did down here. But this is that, and that is this. We have vesicles inside. Out of curiosity, does anybody remember from what organelle vesicles tend to come from? The Golgi apparatus. And what is the structure of a Golgi apparatus made up of? A phospholipid bilayer. That's why the vesicles confuse with the membrane, because they're made of the same material. And they actually become a part of the membrane with that. And you gotta really know that, because it'll help you with the rest of 241 and 242. So to help you with this, because we're going to come back to this a little bit, not tonight, is this, right? These terms here, right? A, you should really already know from cell biology, right? Plus this, right? This I'm going to talk about. But this and that is for you to fill in intentionally, not because I'm lazy, because I love to talk, but because I'm going to make you, if you have not yet read that damn sheet on Canvas called The Summary of Cell Biology, and from there you'll find the answers to this, and do it exactly how I typed it out. Right? Because it'll help you make sense of the rest of 241 and 242. If I get you to master those 5 or 6 pages, 241 and 242 are a lot easier. I just keep saying the same thing over again, over and over for the next 2 quarters. I just change the scenario. Heart cells, muscle cells, digestive tissue, an endocrine gland. It's the same concept over and over and over again. So do tomorrow, learn those 4 sheets. Don't memorize them, understand the principles behind them. Because then, like the idea of when chemicals bind to proteins, whenever we talk about a chemical, that little red dot's a chemical. When it lands on that gray thing, the gray thing changes shape and allows those purple and blue things to move through them. When the little chemical comes off, that gray gate closes and those things can't move through it anymore. That's how we control the diffusion of ions across the cell membrane. But you also want to keep in mind the idea of net diffusion, facilitated diffusion, and simple diffusion. All right, and I'll make sure you know them on Wednesday in that sense. But you should walk into Wednesday going, yeah, I got these down, dude. And active transport compared to diffusion, right? And osmosis compared to diffusion. Oh my God, right? All these things we expect you to remember. All right, I am done. I'm shutting up. All right, if you guys are adults, do what you need to do. You can either say, dude, I'm totally brain dead. It's time for a couple of shots of bourbon and a couple of beers, right? Or I'm going to work on my muscle list, whatever it is. I will trust you guys to make the appropriate adult decision based upon your certain circumstances and scenario.